Okay, so let's dive into this really amazing story about how AI is learning to read the book of life in a completely new way. We're talking about the code that literally makes us who we are, the human genome.
Just let that number sink in for a second. 3 billion. The human genome is a code that's over 3 billion letters long. I mean, that's the entire instruction manual for building a person, and it's packed into almost every single one of our cells.
You see, for years, AI models have been trying to just read that sequence of letters to figure out how our genes are controlled, but they kept hitting this wall. Why? Because it turns out, just like with human language, the same genetic word can mean something totally different depending on the context. The meaning can literally change from cell to cell.
So the crucial point here isn't just about building bigger and bigger AI models to brute force the problem. Nope. The real breakthrough is coming from making AI that's smarter and more specialized, an AI that can actually understand that all-important context.
And that brings us to our first big idea. How in the world do you teach an AI to understand the context of the genome? How do you get it to grasp that the exact same set of instructions can build things that are wildly different?
Now this slide right here, this gets to the absolute heart of the puzzle. Just think about it. A brain cell and a liver cell in your body have the exact same 3 billion letter DNA sequence, and yet one is processing thoughts while the other is filtering toxins. They couldn't be more different. So how does each cell know what it's supposed to be?
And this is the exact limitation researchers were struggling with. An AI trained on, say, liver cells just couldn't make good predictions for brain cells because it was missing that vital piece of the puzzle, the context. This is precisely the problem that a brand new model called EpiBERT was built to solve.
The answer, it turns out, lies in something called chromatin accessibility. It's basically a second layer of information that sits on top of the DNA itself. So if you think of the genome as this massive instruction book, chromatin accessibility is like a custom set of highlighters for each cell, marking up which specific pages and paragraphs are important and which ones it should just ignore.
So, how exactly does EpiBERT use the second layer of information? Let's take a look at how it builds a much more complete picture of what's really going on inside our cells.
The way it learns is just brilliant, and it's actually inspired by language models like ChatGPT. The AI is basically playing a biological version of fill-in-the-blanks. It looks at the DNA sequence and most of the cell's highlighting map, but then a little piece of that map is hidden. Its whole job is to guess the missing highlights. And by doing this millions and millions of times, it starts to learn the deep, fundamental rules connecting the DNA letters to their specific job in that specific cell.
And the results? They're fantastic. Because EpiBERT learns from two kinds of data, the raw sequence and that accessibility map, it can generalize its knowledge. What that means is it can make really accurate predictions for a cell type it has never even seen before. That is a huge leap forward. And the best part? It does all this while matching the performance of much, much larger models that only look at the sequence.
Okay, so adding more types of data, like EpiBERT did, is one really smart way to go. But what if there's another way? What if for certain biological problems, we need to design a completely different kind of AI? This is where our second story kicks in.
The AI models that dominate today, things like transformers, are absolutely brilliant at understanding language, and that's because they get that order, or relative position, is everything. Eat fish and fish eat use the same words, but the order changes the meaning completely. Transformers are built to understand that.
But here's the thing. In biology, some data isn't like a sentence at all. Take, for example, a list of all the active genes in one single cell. It's really just a bag of genes. It's more like a grocery list. It doesn't matter if you write down milk before eggs or eggs before milk, the stuff in your shopping cart is exactly the same. The order is totally irrelevant.
Which leads to a really interesting question, right? When we use a super powerful AI that's designed to understand order on data where order doesn't matter, are we using the wrong tool for the job? Is it just inefficient? Maybe even just plain overkill? Well, this is where a different kind of AI architecture comes into play. Graph neural networks, or GNNs. See, they're designed to understand relationships, not necessarily a strict sequence, which makes them a really fascinating alternative for these kinds of problems.
And just look at this comparison. When you put them head-to-head on this grocery list kind of data, GNNs get virtually the same performance as the big transformer models. But, and this is the kicker, they do it using a tiny, tiny fraction of the computational power and memory.
That's right. In these experiments, the GNNs needed only about 1/8 of the GPU memory. I mean, that is a total game changer. It makes this kind of powerful biological analysis way more accessible, way more sustainable. It means more scientists can run these experiments without needing a massive supercomputer.
So when you put these two very different stories together, the story of EpiBERT and the story of GNNs, what does it all tell us about the future of AI in biology?
It shows us that the future isn't about building one single, giant, all-powerful AI to rule them all. No, it's about building a diverse toolkit of specialized AIs. EpiBERT teaches us to get smarter by adding more types of relevant data for context. And GNNs teach us to get way more efficient by matching the AI's core design to the actual nature of the data we're looking at.
And that right there is the big takeaway. Specialization is absolutely key. It's what leads to more efficient, more adaptable, and ultimately way more powerful tools for understanding disease, for discovering new medicines, and for continuing to unravel the secrets in the book of life.
And it leaves us with this final really exciting thought, you know? Chromatin accessibility was one hidden layer of information just sitting there. How many more are there? What other secrets are hiding in our genome, just waiting for us to build the right AI key to finally unlock them?